Very much to the organizer for giving me that opportunity. So we're back to the field of devices, implantable devices like in interventional cardiology, neurology. So you may think of stents, valves, occluders, everything that goes in contact with the blood inside the body. I'm proud and happy to share, I would say, the team is working with me around this project, and special thanks to Professor Giuseppina Caligiuri, who is the co-inventor of this technology I'm going to present to you, and to Professor Patrick Sirois, who is supporting us very strongly on this journey. The problem we are trying to tackle comes from the observation that the history of interventional cardiology is that of a post-intervention thrombosis control. Whatever good device we do, 30 to 40% of PCI patients are at high risk of bleeding. The very large number of randomized trials which are still running in order to increase, decrease, adjust the antiplatelet therapy that is accompanying the thrombo-inflammation following the implantation of the device are here to testify that. The bad news is that there are no biomarkers for high bleeding risk. So now there is an enormous amount of data which ends up being a very well-educated guess for cardiologists when they have the patient in front of them on the table. And the second issue is also that the incomplete definition of healing. We have in the field of interventional cardiology and neurology always considered that the topological coverage of devices by endothelial cells that are connected and releasing nitrogen monoxide is the indication that I would say the healing is being done, and this is probably only 50% of the truth. We are still making fully covering endothelial layer, which are pro-inflammatory, on which we see fibrin on them, which is the indication that actually they have not restored their physiological phenotype. So what we have decided to try is to actually find a solution to the problem at the device level so that it would be patient independent. And we've been working essentially at going back to why does our blood not clot in our arteries, in a healthy artery, to start with. And you all know that leukocytes, endothelial cells, platelets, they all have one big point in common, is that they all have CD31 on their surface. So this transmembrane protein, which is having 6 domains, 6 immunoglobulin domain on the outside, and a part inside the cell, which is actually, I would say, piloting their item motifs in order to start a certain number of dephosphorylation reaction. And the recognition, called the trans-homophilic interaction, you can imagine upstairs like a platelet and the one downstairs is an endothelial cell. This is the way they recognize, by making this interdigitation, which essentially the net result is that platelets will bounce on the surface of endothelium like a balloon would do. So we have decided to try and borrow from the CD31 peptide on the last 2 domains, domain 1 and domain 2, graft them on the surface and see whether it could be enough to bluff the platelets and the leukocytes and make them believe that the surface of the device is actually an already healthy endothelium that is readily formed. In such a way, we camouflage the device. This is the reason why you see this beautiful animal on the right, which is having the possibility to change its color and be quasi-invisible to the outside world. So the first thing we did is that we tried to identify small peptide, less than 20 amino acid, because we wanted to make chemical synthesis and not biotechnology synthesis. And second criterion that we have been using is that we wanted to give a meaning to animal trials. So we have tried to find consensus sequence of CD31 that would be the same for humans, for rabbits, for pigs, all of the animals on which we would make some tests. And last but not the least, we were trying to identify which peptide was actually having the best impact on the trans-homophilic interaction. So you see here 5 of our candidates that we have tested, essentially selected in the beta sheet regions of the interaction between the domain 1 and domain 2. And what we do is that we make coatings out of them, and in such a way that they are available to the interaction with some endothelial cells, UVEC endothelial cells, that we culture on the surface. And then we compare bare metal hydrophilic polymer, which is usually the, I would say, benchmark in everything that is supposed to be reducing thrombosis, and our CD31 coating. And what we measure is the activity of the CD31 of this UVEC compared to the activity of their actin. So actin revealing a stress of the cell and the fact that the interaction is, I would say, non-natural. And by making the ratio between the two, we defined a dimensionless parameter called the physiological index. Closer to 1 is actually better, meaning that actually we have indeed bluffed the endothelial cell, the human endothelial cell, with our coating. And what you can see here is that first, bare metal and polymers, even though they make full coverage, because in this kind of study we can fully cover the surface with the endothelial cells, their physiological score is pretty low compared to the one we can obtain with CD31. And you can see also with the graph on the right that all of the peptide candidates we have tested do not necessarily rate in the same manner. So the one that wins the game is group 2. So if I go back to here, it is essentially this one, SP1072, which is winning the game as to having the biggest impact directly on the transhomophility interaction. So we've been using that, and the purpose of what I'm going to show you now is just to see how far are we bluffing. So if you know the pathway of CD31, it can do plenty of things, reduce inflammation, whatever. So this we do not invent, of course, it is already known to the literature. What we show here is the extent to which our small peptide on the surface can really mimic the whole of the CD31 pathway. The first thing it can achieve is that we clearly see when we compare on a balloon expandable stent, BMS compared to drug-eluting stent, this is sirolimus in that case, at 30 days, we see that a CD31 layer alone, number one, builds an endothelial layer. Second, it's an endothelial layer which is probably non-pro-inflammatory, because we see none of the adhesion of platelets and leukocytes that we can see on the other two. Picture on the right is also showing you something that we're not very much used to, is to see an endothelial layer with no fibrin on it and no sign of leukocyte. So there is no sign of inflammation relating to this. At 30 days, it's a bit, I would say, too early to tell whether we are fully inhibiting restenosis, but as a matter of fact, we will see later, we have some data showing that not only we reduce the inflammation, but actually we just stop in its track the whole inflammation cascade, eventually leading to the restenosis without any need for any drug. A second test we have done was on a self-expandable flow diverter type of thing used in the brain. And then the same story here, but we are only at 7 days. And what we see is that we have a full endothelialization, again, without any sign of platelet or leukocyte adsorption, only at 7 days. Another test that we have done is that we've been following that at 28 days, and without a drug looking at 28 days makes sense from the neointima standpoint. And what we see here in the case of CD31 is that we have no neointima. So this is normal for a true CD31. This is normally what it should do. There is no reason to have neointima proliferation on top of a, I would say, fully physiological CD31. What we show here is that our peptide coating actually does the same. So it reproduces pretty nicely the natural mechanism of reducing the inflammation and cutting the thrombo-inflammation cascade. Last but not the least, we have examined situations of a flow diverter which is used in an aneuritic model. So we make an aneurysm in a rabbit, we do the implantation of the flow diverter, and you see two things. So first, the pink column is essentially showing the same as before, no neointima. We also see that there is no inflammation, in particular close to the aneurysmal sac. More interestingly is the picture with the yellow circles. You can see that at 60 days, we have totally closed the entrance of the aneurysmal sac. So there has been a full reconstruction that has taken place at the entrance of the aneurysm, which is something which is notably difficult to happen, and usually what people cannot achieve by just having, I would say, a clot forming inside the aneurysmal sac. If we make, I would say, a closer glance, this picture to me is extremely interesting. So the gap between the stent and the tissue is just the result of the forming of the slide. But what is the most interesting is these layers that you can see. This is not a clot anymore. These are layers of extracellular matrix. So it means that the clot is fully stabilized. There is no blood circulating inside, so no possibility to rupture this aneurysm. And it shows that in this area, centimeters away from where the action is, meaning the surface of the stent, the inflammation has been reduced in a sufficient manner to actually trigger the polarization towards M2 macrophages that are making the full reconstruction. So if we are continuing to work on that, as I was telling you, all of this is just the normal CD31 pathway. We already know that CD31 can do that. What was not obvious and that we are progressively demonstrating is that having a coating on a device in contact with the blood is not only going to limit the adhesion of platelets, but it is also going to propagate a lowering globally of the inflammation. Everybody chill down with all the benefits that can go with this one. So this is the takeaway. I'm a bit out of time, I guess, so I'm not going to go through each and every point. I guess you have understood. If ever, because I went a bit fast, you want to download this presentation, you can do so just by scanning this QR code. Thank you very much.